What I'm going to talk about is not cell therapy. This is obviously cellular therapy is a critical part of the new emerging armamentarium of how we can treat cancer with various immune modulations and manipulations. But I'm going to talk mostly about what we've learned concerning the mechanism of checkpoint inhibitors, which really, is kind of the big thing in terms of how most of us treat patients with a wide variety of indications now. I should start by saying that I will soon be leaving Genentech because despite all of the successes that the field has had, and actually the group has had with respect to pushing out at least now 2 rather major drugs, the corporate powers that be have decided to de-emphasize the area, and they took apart the department, which I'll come back to in a bit. And so I decided I'll do something else. So that's still a work in process, but that will happen. So as a result, I kind of feel obliged to start with this. It really is the case that everything has changed just over an incredibly short period of time in terms of our understanding, not only of patient care with respect to in oncology, but also just understanding cancer biology itself. I mean, the original hallmarks of cancer that we all know and love from Doug Hennahan and his colleagues actually did not consider the immune system or even the tumor microenvironment as part of the hallmarks of cancer when it first came out. That's now been corrected. But nevertheless, that kind of sums up why such a big change has been made. So 10 years ago was the first approval of a checkpoint inhibitor that blocks the PD-1, PD-L1 pathway, which of course is the most important and efficacious pathway to block. And that was with the approval of nivolumab from Bristol Myers. This, I think, engendered a complete change in oncology practice, and as I said, a change in our understanding of cancer biology, and it was really kind of the rebirth of cancer immunology as a field, because up until now, although it's been around for a long time, and those of us who have been in the Ludwig Institute certainly know that very well, it was kind of in the doldrums because the work was not very impressive, shall we say, and certainly the clinical results even less so. But to give you an idea just how much things have changed last year, the aggregate of immunotherapies, actually just the checkpoint inhibitors, resulted in $70 billion of sales worldwide. And I think for maybe the first time in history, an oncology drug, Keytruda, was the world's best-selling drug. So you would think that this would really be an important thing that would drive further investment in the field. But 10 years has gone by, and another meteor hasn't fallen, and so in the way of how many big pharma companies think, and those of you who think Genentech is a biotech company, you were wrong. It's been embraced warmly by the Roche organization, which actually is a great company, but nevertheless is subject to these kinds of decision-making practices. So what do we have, though? In fact, over the last 10 years, there's really been an incredible rise in cell-based therapies, both for cancer and autoimmunity. And Stanford, certainly with Crystal Mackel's group, has been in the vanguard of pushing these things forward. There have been a whole bunch of bispecific antibodies that have come out. One from our group, which is mosunetuzumab, which is a anti-CD20, CD3 bispecific that's used in lymphoma in increasingly earlier lines of therapy. And I think we're now seeing a return of something that really is a zombie, and many people would have preferred to remain dead, which is cancer vaccines. I'll share a little bit of our own data on this point, but this is certainly something that I think we all have to keep our eyes on. So other thing to keep in mind that we're looking here not just at individual therapies, but really a whole new modality. and a whole new way of thinking about how we can treat and attack cancer that is quite orthogonal to almost everything else that's been done so far. But be that as it may, I think the future will be in how you combine one type of targeted therapy together with an immunotherapy, because you can take the attitude that anytime you do anything to a tumor, be it liquid or solid, you're also altering the immune response to that tumor, and understanding that interaction, and understanding the cells that play a role, and which ones lend themselves to therapeutic intervention, is something now that there's such incredible proof of concept, something that I think bears more rather than less investment. And I think fortunately, academic institutions are really stepping up big time in this area. And with the exit of large pharma, or I shouldn't say exit, but at least the withdrawal of as much enthusiasm in favor of weight loss drugs, the VC community is starting to pay more and more attention. So I wasn't quite honest in saying I never come down here. I've been down here a lot now, actually talking to venture capitalists very close by here, and they seem very enthusiastic about trying to fill the gap, understanding that the pendulum will swing back, and then everybody will want one. Some years ago, Dan Chen, who actually is a Stanford graduate, and I decided to try and put some simple sense to understanding what has to happen in order for the immune system to mount a response to cancer. And this gave rise to something we didn't think was going to be that popular, but turned out to be, and this is called the cancer immunity cycle. And it just, for those of you who don't really think about these things very often, it's kind of useful. And it was also very useful to us to understand that if you think of all of the steps that occur in cancer immunity as cyclical in nature, then you know that any one step at any one time is going to be rate limiting, and that gives you an opportunity to try and understand what is that step, and then what happens if you attack that step with a particular therapeutic. So just briefly, just to put everybody on the same page, you have a tumor, and the tumor is invaded, hopefully, by antigen-presenting cells called dendritic cells. And these cells are very astute and efficient at being able to generate T-cell responses. And they do so by eating dead tumor cells, cleaving them up into peptides, and presenting them on MHC class 1 or class 2 molecules to T-cells that they encounter after migrating up the afferent lymphatics into draining lymph nodes of the tumor. T-cells then are stimulated here. They exit into the bloodstream and then find their way to a tumor, where they then extravasate, enter the tumor, and supposedly recognize the cognate peptide MHC complex is on the tumor cell, and then kill it, starting this whole thing again. Now, this whole process is countermanded in most patients by the phenomenon of T-cell exhaustion, which I'll come back to in a moment. And this means that the tumor, either advertently or inadvertently, is able not to kill, but to reverse or attenuate the function of T-cells by causing them to go into a chronic quiescent state, which actually also happens and was indeed first defined by the behavior of T-cells during the course of a chronic virus infection. The T-cells are not dead, but they're not as active as they once were. So that gave rise, now some time ago, in the mid-2000s, I said to the idea that, if we could reverse T-cell exhaustion or block it from occurring in the first place, we might have a therapy. So that's what gave rise to the whole idea of these so-called checkpoint inhibitors, because it is a particular checkpoint, particularly the PD-1 molecule, that seems to control this exhaustion pathway. And in a moment, I'll tell you how we think this works. But I also want to tell you that we realized that first incarnation was a bit simplistic, not that it's untrue, but that what faces a T cell after it migrates out of the blood into the tumor is a good deal more complicated than simply finding the tumor cell and killing it. the T cell has to negotiate its way through tumor microenvironment, particularly tumor stroma, and overcome a variety of different immunosuppressive mechanisms that it can encounter there. But also it encounters more dendritic cells coming in, which in fact help the T cell actually try to do its job. And this gives rise to the formation of what has been seen for years, but now are starting to be understood from a mechanistic point of view, these so-called tertiary lymphoid structures, which are like isolated lymph nodes with germinal centers that one finds scattered in and around tumors. So that's a positive thing, but there's a lot of negative immunosuppressive features that take place. One way of illustrating that is shown here. Again, a highly simplified view of life, but nevertheless, I think one that's been very useful to us in plotting our own directions. We like to break down tumors as having one of three different immunotypes. So you have genotype, you've got, and you also have immunotype, and this is how a tumor reacts to an immune response. So on the left, you find tumors that most of us will simply call hot. These are tumors that are, we call them inflamed tumors. They're very much filled with T cells and other cells of the immune system that you don't see, most notably dendritic cells. So when you do single cell or even bulk sequencing on these tumors, you find lots of active T cells. In the middle is what we call the immune excluded tumor, and bear in mind all of these things occur on a continuum and are also probably dynamic in nature. But nevertheless, the immune excluded group are patients who have a T cell response that you can monitor and demonstrate objectively to their tumors, but the T cells can't get to where the tumor is because of a rather significant stromal investment that occurs in and around the tumor that actually physically restrains the T cells from entering the tumor nest. And finally, the immune desert, where there's nothing going on. And often we find these tumors associated with a distinct neuroendocrine tumor, the endocrine signature that is not only found in neuroendocrine tumors, but can be found anywhere. And what this seems to do is keep out the dendritic cells somehow. And so absent dendritic cells, nothing has a chance of happening. I don't know that that is the original sin in these tumors, but it's certainly something that we're all very interested in. On the bottom, I'm just showing you that all of these phenotypes are rather prevalent. And in non-small cell lung cancer, which of course we view as one of the tumors that's highly responsive to immunotherapy, only about 1/3 of the patients have the immune inflamed phenotype, and it is that phenotype or immunotype that is the one that is most highly responsive, has the highest probability of response to checkpoint inhibitors. About half of this patients actually have evidence of these immune excluded tumors. By my estimation, if you have an immune excluded tumor, you're not going to be very responsive to a checkpoint inhibitor. So this may indicate one reason why it is only about 25 to 35% of non-small cell lung cancer patients actually respond. They will never respond unless you do something about the barrier to T cell action. So what I'd like to go through now is as quickly as possible, so as not to take too much time. Three things. First of all, how does checkpoint inhibition work? This is something that remarkably few people in the field have really taken on seriously, but I think it's important to know why things work when they work in order to understand why things don't work when they don't work, which is a far more common situation. Insights into how T cells develop and how all of this is controlled by checkpoint inhibitors, and then also highlighting what I think is the emerging importance of dendritic cells in controlling all of this. Now, T cell exhaustion is something that's been around for quite some time as a concept. I think it was first introduced by Nick Ricifo at the NCI, but more recently, people like John Wherry and Andrea Schiedinger and Dietmar Zen have defined what we mean by exhaustion. And it seems to be a progression of events that take a perfectly good T cell precursor, when exposed to chronic antigen, as one finds in the case of a tumor, these T cell precursors, which sometimes are called TPEX for exhausted precursors, are gradually modified epigenetically to become a fully exhausted T cell. And by the time you get to that point, in fact, there's no turning back. These cells are so heavily locked into this particular exhausted, suboptimal, grossly suboptimal phenotype from a tumor killing point of view or virus killing point of view, that they can't come back. And although I think we thought that the way checkpoint inhibitors initially was going to be very simple, these exhausted T cells are high in PD-1, which is the negative regulator, the co-inhibitory molecule or checkpoint. Many different terms can be used to describe them. So we assume that inhibition of PD-1 would be presumed to reverse exhaustion. But as I said, these cells are so locked in an epigenetic state that, in fact, they don't go back. So it gets you to reevaluate what is it that PD-1 is. Well, it's also a marker of early activation. And I think that gave rise to the possible mechanism, which I think turns out to be correct, that indeed PD-1 activity drives exhaustion. But actually doesn't do it directly, as I'll show you in a minute. Now, these are the two major signaling pathways that are relevant in T cells. First, in the case of the T cell receptor, which recognizes its cognate peptide bound to either class 1 or class 2 MHC molecules. TCRs generally will signal via the RAS MAP kinase pathway, at least in the first instance. But in order for a T cell to become fully active, you need a second signal. These are called co-stimulatory molecules, and probably the most famous of these is CD28. Which binds ligands that are present particularly on immune cells, such as CD86 or CD80. And CD28 signals predominantly, again, in proximal signaling mode via the PI3 kinase AKT-mTOR pathways. You've got two parallel pathways that are going on. They even obviously interact. But nevertheless, these are the two molecules that are needed. PD-1 sits in the middle here, and all we knew about this coming in is that it has a inhibitory domain in its cytoplasmic tail, which recruits one or more phosphatases, such as SHP2 or SHP1. So what were these phosphatases doing? Summarizing a lot of cell culture work and vivo work, as well as in vitro reconstitution of these pathways. What we found is what's shown here is that activation of the T cell by the T cell receptor winds up phosphorylating the cytoplasmic domain of PD-1, which creates a binding site predominantly for the phosphatase SHP2, which is recruited. And then the big surprise was, rather than having SHP2 dephosphorylate the T cell receptor itself, it actually looks in the first instance towards the CD28 co-stimulatory molecule. There's lots of other stuff that regulates T cell receptor signaling, but and in fact, SHP2 will do this too in the absence of CD28. But all of the data that we and others now have really suggests that CD28 is a very important target for that. We can skip that. And then started asking the question, well, how do dendritic cells or other myeloid cells fit into this? I think the most important thing to bear in mind is that most tumors, certainly most solid tumors, do not express ligands for CD28, such as CD80 or B71, B72, which is otherwise known as CD86. So what that means is that when if you're imagining then that the way exhaustion works is that when a T cell comes into a tumor, if it needs CD28 signaling to work, the tumor is not going to be supplying the ligand for CD28. Something else has to. So we're hypothesized that that was going to be the myeloid cells, which means either macrophages, which are quite numerous in mouse and human tumors, or dendritic cells, which are the cells that are actually most potent at presenting antigen that gets the T cells off the ground. Which is it? And this is an experiment done some time ago now by Soyoung Oh, in which he knocked out the PD-L1 gene from tumor cells, from the germ, from all cells in the mouse, or just in dendritic cells. And she found that PD-L1 expression by tumor cells really didn't matter that much. The germline knockout shows here that when you eliminate PD-L1 expression in the mouse, the tumors don't grow. That's what all these gray traces are. But if you knock out PD-L1 only in dendritic cells, you get pretty much the same result, which means that of all the cells in the body, the only relevant population of PD-L1 that contributes to the exhaustion or inability of T cells to control the tumor is the PD-L1 that's expressed by dendritic cells, and to some extent macrophages as well, but certainly dendritic cells are punching well above their weight. Now, there's a number of other of these co-inhibitory molecules, one of which now actually has an antibody that has received FDA approval to something called LAG-3, which may actually work in somewhat of a different fashion. But we got interested early on in this molecule called TIGIT, in part because it was discovered at Genentech long before I was born, but nevertheless, it was there, and so all the reagents and a good knowledge base was there. What TIGIT is it seems to have the same role with a second co-stimulatory molecule called CD226 that PD-1 has with CD28, namely TIGIT inhibits CD226 activity. It does this predominantly by competing for ligand. CD155, or the poliovirus receptor, is one of the primary ligands for both TIGIT and CD226. And if there's TIGIT around, there's no ligand available for CD226 to bind. So the therapeutic hypothesis then becomes, well, okay, let's make an antibody to TIGIT, block its ability to bind the ligand, and then everything will be fine. In fact, initial clinical results made it look that way. This is a randomized phase 2 study showing that a combination of an anti-PD-L1 antibody called atezolizumab together with tiragolumab, which is the anti-TIGIT antibody, in non-small cell patients actually elicited a quite impressive overall survival benefit. Also a PFS benefit, but we were much more interested in the OS benefit here. And this is particularly so in patients who have these immune inflamed tumors. The development group then started putting this in all sorts of other tumors that we had no evidence that really this was going to work. And perhaps not entirely surprising to me, at least, there have been some notable phase 3 failures in this. We're still waiting, hopefully by the end of this year, for a trial called Skyscraper 1, which is the actual only trial that was planned that actually repeats the phase 2 experiment. So we're, I'm obviously nervous about this, but we're anxiously awaiting and still optimistic that that readout will give us something. It had a positive PFS readout, but we ignored that. Because we didn't feel that that was going to be a viable option for patients. Now, so how does TIGIT work? We did the same sorts of experiments with TIGIT as we did with PD-1 and found, to our surprise, that the PD-1 pathway and the TIGIT pathway really converge to provide one element of control to T cells. And this is both an advantage and a disadvantage. In other words, we're still, by inhibiting TIGIT, inhibiting the same pathway that we're inhibiting with PD-1. In other words, CD226 also signals via the PI3 kinase pathway, but it's the PD-1 cytoplasmic domain that still recruits SHP2 and dephosphorylates both CD226 and CD28, cutting off even more effectively co-stimulatory molecule signaling. The role of TIGIT itself seems to be more just in regulating this initial ligand binding step, which is important, but to get full activity, you need really to block both of them. So what are the functional consequences of this? Now, I'll dip into a little bit of immunology. If you I want to show you data from the combination studies. These are now mouse studies that if you block with the combination, you see a drastic increase in the number of T cells, and indeed those T cells are tumor specific. And we can tell that using a tetramer to a particular tumor antigen in this mouse model. But as an interesting, what you find is a decrease in a transcription factor called TOX. Now, what TOX is, for those of you who don't know, is one of the early transcription factors that initiates the exhaustion pathway in T cells. So when T cells get overstimulated, TOX goes up, and the cells then start committing to exhaustion. If you can keep TOX down, then presumably you can avoid exhaustion. Armed with this, actually my group of postdocs went and did an enormous single cell sequencing experiment, dividing up the constellation of T cells that one finds, this is all in mouse tumors, and analyze them not only in terms of what they express, but in terms of what their functional activities were, and what antigens they saw. In other words, were they or were they not tumor specific? I won't take you through all the single cell heat map, but rather go to the data that contains the conclusions. So here what you're looking at is an experiment that really hadn't been done before, which is to harvest enough mice so that you can actually measure what happens to T cells in the draining lymph node, what happens to T cells in the blood, and what happens to T cells in the tumor. And what you find is that, again, you find T cells that are expanded in the tumor, and the x-axis T cells that are expanded in the draining lymph node. And adding anti-PD-L1 alone has an effect. Adding TIGIT alone has an effect. But when you add them both together, now you find a rather marked expansion of T cells, both in draining lymph node and in the tumor. And by the size of these circles here, you also find them in the blood. Okay, so presumably they're being made somewhere, they spill into the blood, and then go along their merry way. So in addition, all of those cells in the blood, we can show based on T cell receptor sequence analysis and functional analysis, those cells as shown here by the purple colors are all tumor antigen specific. So they're not just any run-of-the-mill random T cells. In fact, these are the T cells that you would like to see made and are most heavily elicited by the combination. You look at, this is a kind of T cell trajectory analysis showing you what happens to T cells in these, at least in lymph node and in tumor when you do nothing, or when you add either agent alone or either agent together in combination. These are the TSCM cells in control animals are the precursors for all T cell compartments. They're called the T stem cell memory compartment. And you find them in lymph nodes going, starting here and then entering an effector type compartment, which we don't entirely understand. But in the tumor, you find that almost all of the cells progress to the exhaustion phenotype, which is shown here at the bottom of these UMAPs. But when you treat these mice with combination, the situation changes really quite a bit. These TSCM cells enter into a new compartment, which we call a CCL5. And those CCL5 cells, which you can track based on the unique sequences of the T cell receptor found in each of these cell types, then appear in the blood. And then these same CCL5 cells also can be shown, again, by this TCR lineage analysis to enter the tumor, where they then radiate into effector cells. And as you can see here, the exhaustion pathway is almost completely cut off. So this is a really effective way at keeping T cells happy, so to speak. And this is a diagram that kind of presents the working model for all of this. We imagine that because dendritic cells are important for initiating T cell responses, that they're the cells that are responsible for generating this TSCM compartment in the first place. We know this compartment, although I didn't show you the data for it, is expanded as a consequence of these inhibitors, but in any event, they give rise uniquely to these two different CCL5 type cells that are called transit cells, or these are the TPEC cells. I'm not really too concerned about what you call them, except to say that all of the T cell clonotypes that you find as having been elicited by the checkpoint inhibitor are found in these transit cells, the CCL5 cells, which then enter the tumor, encounter dendritic cells again, presumably, and then differentiate either into memory cells, T effector cells, or T exhausted cells. And the important feature here is that if you block PD-L1 or PD-1 and block TIGIT, you cut off the exhaustion pathway. And I think the best hypothesis for why that would happen, and in fact, I'll show you a bit of data for that in just a moment, is that you favor exhaustion when you have insufficient co-stimulation. Okay, it's and then all the things that most of my colleagues study who were that don't think that anything important happens in T cells. It's not a transcription factor. All the transcription factors are getting their marching orders from the signaling patterns that are happening in the periphery, and we would maintain that all of that stuff is set up in the first instance by what is the balance between signal one, in other words, the T cell receptor signal, and the co-stimulatory signals that occur. If that's the case, then indeed that explains at least one aspect, the T cell centric aspect of how these checkpoint inhibitors work. This just shows by correlation, if we go back to that Cityscape phase 2 trial that I showed you that nice result for, if you go back and look at patients and ask, is there any evidence in the blood of any of this going on, best I can show you is that those patients who respond best actually have a lot of these CCL5 cells in the blood. And if that's, so that would be consistent with saying, okay, the dual inhibition elicited a lot of these cells, and you can see them in the blood. But again, this is just an association and certainly not a mechanism. Now, I showed you that one knockout experiment to suggest or indicate that in the PD-1 pathway, dendritic cells are important. Question then, is that also true in the case of TIGIT? And the answer to that question, just to cut a long story short, is yes. If you knock out dendritic cells entirely from mice, you find they lose their ability to control tumors when given the combination treatment of TIGIT plus PD-L1. Is that due to the expression of the ligand PVR on dendritic cells? The answer to that question is yes, because here Ellen Duong has selectively knocked out PVR from dendritic cells and basically gets the same type of result that So-Young Oh had received earlier, namely that here you can see there's been a increase in the amount of tox that occurs, and that's knocked down with the combination treatment. But if you remove PVR, the ligand for TIGIT, from specifically from dendritic cells, then you find that the combination treatment loses its ability to reduce tox levels. In terms of tumor control, you get more or less the same result. It's certainly not as impressive as we saw with the PD-L1 knockout. And that's probably because PVR, unlike PD-1, is very, very widely expressed in tumor cells, non-tumor cells, stromal cells, everywhere. So the antibody is going to be more efficient than the knockout in that sense. Now, just to show you that both pathways are important, this is the experiment I promised you, is that if you use an antibody to block CD226 signaling or to block CD28 signaling, this antibody ties up the ligand, this antibody ties up the CD226 receptor. What you find is that the combination therapy loses its ability to control tumor growth, which means that again, it's completely consistent with the idea saying that at this stage, you need both CD28 and CD226 signaling in order to prevent exhaustion and optimize the growth of tumors. Now, just for the last couple of minutes, I'd like to switch gears a bit and deal with this issue of vaccines, which is something that I personally have been like super interested in since my own fellowship period. To give you an idea how long people have been thinking about tumor vaccines. This is still, I would consider to be a way of enhancing endogenous immunity because you're trying to use an approach that will increase the number of T cells that the endogenous immune system can make, say, as opposed to a synthetic approach where you're using either a bispecific antibody or delivering modified cells to patients. So some years ago, I met up with Ugur Sahin at BioNTech. This is before he was famous. Due to COVID, and we hatched an idea together with BioNTech that we could do this by identifying all of those mutations that occur in tumor cells, figuring out which ones are likely to elicit T cell responses, and then make an mRNA vaccine to those and deliver that vaccine to patients, first to mice, obviously, and then to patients. The formulation that we decided to use is not the COVID formulation, but one that's actually more immunostimulatory. This is an intravenous injection of an unmodified RNA. So in other words, it's also a very good adjuvant for the immune response. It binds to TLR7 and TLR9 or TLR8 very well. So we deliver these lipid RNA complexes intravenously. They go to the spleen. They enter dendritic cells and macrophages in the spleen, particularly the dendritic cells, then make the antigens and present them to T cells. So you're again using dendritic cells as the way of doing a vaccine. The prediction of the immunogenic or antigenic epitopes is an important element of this. So here, Kai Lu and Sushit Junjunwala in the group, two computational biologists, terrific ones at that, developed a machine learner which gave rise to an AI, which is actually now in the clinic. And I think it may be one of the only AIs that's actually doing something like this as an integral part of a therapy. So this is an approach called giraffe, where we computationally predict those peptides that are most likely to be immunogenic, and then we encode those into the mRNA, and those are the mRNAs that are then given to patients. We went through a couple of studies looking at metastatic disease, particularly metastatic melanoma, found we were getting really good immunizations. A paper on this is now, I think, in press in Nature Medicine, but very little in the way of efficacy. Ugur and I and our groups together with a terrific young pancreatic surgeon, Vinod Balachandran at Memorial Sloan Kettering, found each other at a meeting and thought a better way to test this would be in the adjuvant setting. Even in pancreatic, which of course is never really thought of as being a indication that is susceptible to immune attack. So we figured it wouldn't work, but we'd know real fast because these patients progress so quickly. So in the first set of studies that were done, Vinod himself did all the Whipples. Then took those tumors, designed a vaccine adapted for each individual patient, and then treated those patients both with checkpoint inhibitors as well as with then subsequently the vaccine. And this is all followed up by a standard of care FOLFIRINOX treatment. Now, the patients subdivided unfortunately into two groups. I am not a surgeon, and I've never seen a Whipple done, although I know who Whipple is. My father-in-law studied with him in Rochester. About half the patients, at least in Vinod's hands, have to have splenectomies as a consequence of the surgery. And that's a problem for an IV vaccine because when you inject something intravenously, the first place it's going to be really encountering immune cells is in the spleen. And so people who have splenectomies might have some difficulty making responses. So indeed, that turned out to be the case. But even in this really small phase 1 study, it yielded a unanticipated important feature. Which is that there were half of the patients on the study, about 18 patients, half of them responded to the vaccine, making very nice T cell responses that we've characterized in great detail. And half of the patients did not respond. They did not have a T cell response. Those patients who did not respond all showed regression. Many of them are still alive, but they nevertheless they showed regression within 13 to 14 months, kind of as expected. Whereas those patients, this is now an old data cut from last year, who did respond, none of them at that particular point in time had recurred. We had one patient who developed a lesion on her liver, which we then went ahead and biopsied and sequenced. First of all, found there were no tumor cells there, but it was just all filled with T cells. You sequence the T cells. It turns out that those T cells have the T cell receptor clonotypes that had been elicited by the vaccine. And if you sequence deeply there, you can find that the p53 mutation that was pathognomonic of that patient's tumor could be found there. So this looked like immune surveillance at work, which we'd never really seen before. In other words, a tumor likely seeded itself in the liver, and then it was recognized after that fact by the T cells that then cleaned up. We have a paper we've just resubmitted on a 3-year follow-up. Basically, the situation hasn't changed very much, except that 2 patients who had responded have recurred, and one of those had died. And it turns out that both of these patients had kind of dodgy histories. One, because all this was taking place during COVID, one missed a whole bunch of injections, and actually both did. And as a result, they did not make very good T cell responses. And probably the best way to look at that is here. So here are 6 patients who did respond and did not recur. And here are the 2 patients who did recur. And what we plotted here is the cumulative exposure these patients had to what we would consider to be effective T cell responses. So you can see there's still a large range here, but nevertheless, the T cells in patients who did well have a rather longer lifetime, an average lifetime of 6 to 7 years, as opposed to the T cells in the patients who did recur, which were a year or less. Now, last slide I'll show you about all this is what happens long term. This is 3 and 4+ years out for individual patients. Again, for those patients who continue to do well on this therapy, the clonotypes that were elicited by the vaccine 2-3 years ago all now are found to reside within memory-like compartments that you can estimate will probably outlive the patient, so to speak. In other words, they're there, they're not going anywhere. We anticipate, or at least hope, that these individuals will continue to have their tumors controlled and will live otherwise normal lives. So I think why don't I can have some more information on CD4 cells versus CD8 cells in these responses, but let's skip that and just come to a summary, which, just to remind you, checkpoint inhibitors act to control the exhaustion trajectory by enhancing co-stimulation in a fashion that appears to be dependent on dendritic cells. And that dendritic cell targeted vaccines may provide another approach to cancer, which may very well yet turn out to be transformational, certainly if these early studies in pancreatic cancer, which are now in randomized trials that we are doing at Genentech together with BioNTech, obviously with Vinod as the lead investigator. If those pan out, then we'll have an approach to pancreatic cancer we could never have anticipated. But I also want to leave you with a note which is both cautionary, but also I think very exciting for the future, which is immunotherapy is not just about T cells. There are lots of other cells that the T cells have to negotiate with, as I've said already, in order for them to do their jobs, and that are out there to try and modulate T cell responses. And again, just to remind you, these different immunotypes that we've defined over the years support T cell activity to a greater or lesser extent, even if that T cell activity is being supplied by adoptive cell transfer. So you can give as many T cells to a mouse, certainly, as you like, but if you're giving it to one of these so-called immune desert tumors, the T cells don't find their way there. So we still have to figure out a way to overcome that. This is the group of lab people and clinicians and computational people that really were responsible for all of this over the last 10 years. The specific work on TIGIT was really done by Carl Banta, Katie Nooch, Eugene Chang, Thomas Wu, who did most of the computation work, and Ellen Duong. Vaccine work was, again, another huge collaboration, but Vinod and Ugur Sahin and his partner, Özlem Tureki, really were just incredible partners in getting all of this done. As I said, this is the last picture you'll see of these guys, unless I can reassemble them in a new form. This is, some of us got together at a picnic a couple of weeks ago. My dog Toby was very happy with the number of hot dogs and hamburgers that were consumed. And you'll notice we have a kind of one of my, since it's election season, so one of my parting mentions to them is that get even by doing great work, showing people how stupid the decision this was, by wherever they are, whatever they do great work, and everyone will benefit. That's I think the best way to mete out one's revenge. So our new catchword, and you may see these shirts running around since there are hundreds of them, called Rage Science, and we'll see what the future holds for us all. So again, thank you for this kind and illustrious invitation to speak with you today. And if there's time, I'll certainly be happy to take some questions.
Yeah, very impressive. A few years back, some of your colleagues at Genentech published in Nature that TGF-beta regulated T cell and CD8 T cell infiltration into cold tumors. Is it possible to include and modulate that pathway to enhance the efficacy against cold tumors?
Yeah, no, that's a great question, and I hope it's possible to do that. Since that paper gave rise to a rather large program in trying to see what are the best ways, if any, to safely modulate the TGF-beta pathway. I think we're getting close, but that's going to have to be restarted in a separate way. I think TGF-beta1 is the isoform that is probably most important in the tumor context. TGF-beta2 is the one that drives most of the toxicity. So we are narrowing in on blocking either TGF-beta1 itself or blocking its local activation. We do have 2 programs in the clinic to block activation by targeting the integrins that are responsible for that process. And a small company in Boston called Scholar Rock has a similar program, and AbbVie has a similar program. So if blocking activation is, which is what we found we could do most safely, if that turns out to be efficacious in this context, hopefully we'll know. TGF beta, I love TGF beta. It also actually restrains the size of the TSCM compartment. So when you block TGF beta, those cells expand more, which is a good thing. Plus, TGF beta is required for driving the formation of regulatory T cells, which turn off the efficacious T cell response along with exhaustion. And so you have basically three ways to win. You make more of the precursor, you block Treg formation, and you hopefully change the stromal architecture to enable better T cell infiltration. There's over years I've learned there's a big distance between a good idea and a therapeutic.
Thank you for the great talk. Do you have any opinion about phosphatase enzymes as potential therapeutic targets in immuno-oncology?
Yeah, I mean, they should work. I think the one that's furthest along now is an inhibitor to PTPN2. Through my fifth column network, it's not our inhibitor. We played with that for a while. I think it's pretty toxic because PTN2 is expressed by all cells, not just immune cells. We were interested as well in PTPN22, which is more immune cell specific, and papers still come out on that. Genetically, PTPN22 is linked to lupus, presumably because of its activity in B cells. But we have thus far been unable to repeat a lot of key experiments that you can do in mice in human cells. So until someone sorts that out, I think at least that's where those two sit. And there are other phosphatases as well. I have nothing against them. It used to be said that they're impossible to drug, and that is definitely not the case.
Ira, hi, Dave Miklas.
Yeah.
Many years. Thank you for all your publication, open publication of your data from tech. It's impressive. And as a transplanter CAR T therapist, I want to say, what we learned for 50 years. One is conditioning matters. So a lot of your problems are, I think, that you're trying to do one win and you're not considering the entire sequence of events. Two is marking the cells was critical to every advancement we've made. Look what's happened since CAR T has developed, where we can track the cells and see which cells are important. Three is that the lymphodepletion, which is another form of conditioning, is something which you must be thinking about every day. And then here's my question to you, and it's the thought-provoking question. How frustrated are you with your clinical translational researchers in that you really need the mouse experiment? These patients are dying of cancer. And why you're not taking the T cells out, genetically marking them, or knocking out, or doing the exact thing that you would have done in a mouse in a patient in a stage situation has to be frustrating to you. 10 patients would blow the field open.
Yeah.
TILs have been going on since '78. They're still not marked. I mean, it's kind of ridiculous.
Yeah. The level of frustration is more than you know. Especially because, at least in my place, nobody in our clinical group has any real hands-on experience with any of this, that there aren't even any transplanters who are there. Yet that doesn't prevent them from having very strong opinions on what can or cannot be done. So I'm hoping to change all that. So, all I can really say is I've been working very closely now with Nick Restivo, and we're trying to put something together that will, allow a much more dynamic relationship between those two things, including, one idea that I know several people have talked about, which is rather than using mice to screen for T cell enhancements that will improve a CAR or a TCR-T, do it in humans. Mark the cells, do a CRISPR dropout screen and see which cells survive the best in the context of a human trial. And the FDA at least seems to be on board with that. So I'm hoping to get out from under at least one level of frustration. And we'll visit you, of course. This will all be a Bay Area thing.
Ira, quick question. I'm concerned, as you've been, I'm sure, your career, about the downside of blocking T cell activity, because there's good things T cells do, and you can screw up all sorts of things by the kind of technologies. The personalized vaccine development, cancer specific, sounds spectacularly important to me, given the advent of the RNA vaccine technology through COVID. how far along are you in this? Is that what you're taking away to do somewhere else, or is Genentech committed to developing these cancer vaccines, which sound like a spectacular therapy to me?
They say they will continue the studies that are going, and together with BioNTech. There are other studies also that Moderna is doing with Merck in adjuvant melanoma, and they're going also into other adjuvant indications as well. Problem is that these are extraordinarily expensive therapies. And I think those of us who have been working on this realize that once it started to be so that proof of concept was starting to, and I wouldn't say we're there yet, but it looks like it might be there. Rather significant investment in what is the next generation of the technology going to be that will allow you to scale this and make it available to a large number of patients. That level of investment is not being done. And that's another thing I think that in my next life, however long or short it may be, I'd like to try and address, because there are a lot of ideas that could make these therapies much more cost effective than they are.
So there's no common epitope you could pick up pancreatic cancer, for instance, if you share it. You could
Yeah, there is a theory that you can use shared antigens, but generally speaking, those vaccines, and in fact, those T cell receptors have been less, because probably tolerance comes into play. So if you're looking at these, so-called cancer testis antigens, or even the oncogenic driver ones. I know my friend Liz Jaffe has been working on a RAS vaccine for many years in pancreatic. But those epitopes are always going to be restricted to really weird MHC haplotypes, probably because tumor evolution has selected against mutations that are going to be immunogenic because they'll be selected against. So unfortunately, I think to a large extent, we're stuck with figuring out the personalized approach.
Thanks everybody for sticking with us. And I apologize for those many online. We can't get to all the questions. I do want to end with one more question from Dr. Rubenstein, who it was a great idea to invite Dr. Mellman. So I'll end with you. Thanks, Dr. Rubenstein.
Thanks so much for your great talk. Simple question. Yeah, one of the cardinal features of cancer is Warburg metabolism, moving at high lactate levels, and we've encountered high lactate levels in patients in their spinal fluid as being an impediment to efficacy of lenalidomide, which works primarily through T cells. And are metabolic factors such as lactic acid, in your opinion, important in terms of checkpoint inhibitors in certain cancers?
Yeah. Oh God, yes. It's this is all part of. In this case, it's more than just the tumor microenvironment. A lot of these things become systemic, and I mean, there's a emerging interest and important literature just in the relationship between just metabolic alterations, not only organic acids, but even amino acids, as playing a key role in maintaining T cell health. So I think nicest work from people like Sue Keck at Salk Institute and Erica Pierce at Johns Hopkins. The problem is how to address them. In the sense that you can't, there's so many of these alterations that take place, so you can't go after them one by one. You have to, get to the root cause of it. And I think the more you and others can do to try and figure out, what causes the global metabolic alterations and go after that, that probably will be the most effective approach.
Thanks. Thank you.
Thank you, everybody. And Dr. Fetty is going to bring you up our plaque to commemorate this Rubenstein Lectureship. Dr. Mellman, thank you so much for a wonderful presentation and being with us.
Thanks.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.